Okay, welcome everyone. Welcome again to the fourth course of 9 on clinical variant interpretation analysis. This course is titled ACMG, AMP Guidelines for Single Nucleotide Variant Interpretation, ClinGen Guidelines. And as many of you know, I'm Jade Edelstein from Genix Marketing. It's been really great to hear from so many of you on how you've heard about the course and your feedback during the course. Please write to me with feedback. It helps us to improve the course and know that we're on the right track and doing the right things to make this course valuable for yourselves. Mark your calendars. Genix will be exhibiting quite a few conferences this year. They are both in-person and virtual conferences. And the first one is PacBio Roadshow. It's happening next month. It's in Lisbon. Then London Calling. The big one for the year that many of you, I'm sure, have heard about is ESHG. It's the European Staff in our lab come have been sending me through email. Do we need for logging?
Hi everyone, welcome to class number 4. In this class, as we said before, Dr. Ali Tabish is going to provide a lecture on ACMG classification. But in the previous class, we wanted to give you 2 examples of case reports from the clinic, and we only had time for 1. So very quickly, I'm going to show you another case report, and then we are going to go move to Dr. Ali Tabish's lecture. So this case starts with a woman named Nouine. Nouine is 30 years old. She's a female, and she is pregnant. And she came to the clinic because during one of the routine tests during the first trimester ultrasounds, which is the nuchal translucency test, she was found to have an increase. The fetus was found to have an increased nuchal translucency. In addition to that, during the genetic counseling, this is the pedigree that was taken. Nouine so far she gave birth to 2 children. The first child is 6 years old, female, and this child presented with mild cardiac murmur and bleeding, and this was noted during the tooth extraction. Besides that, there was no genetic counseling or workup that was pursued for this child. The second child was a male, 3 years old, without any medical or surgical history that is relevant. Irene and her husband arrived at genetic counseling due to the increase of nuchal translucency, increase in thickness. In every country, a different threshold would be defined as increased. Specifically, in this case, about 3 centimeters is considered to be high and requires or recommended to pursue with an additional genetic test. So in this specific family, the parents decided to go with a single exome sequencing for the fetus as a solo test. Now we'll go live and see how this case look on an exome sequencing in the platform. So this is the analysis screen, in which we have a summary of all the details, the protocol that was performed here, the genetic models that we'll see, the analysis, and other information, including the version set of all the annotation sources, etc. So if I go to the variant dashboard, this is what we will see. Of course, we provided the phenotype in advance, but we can still change that during the analysis. So if you click on the orange bar here and you go to the phenotypes, you can see that we provided the phenotype here is increased nuchal translucency, which is a phenotype in HPO term, which is well known in fetuses. We must say that this is highly unspecific. This could be a random increase of the nipple translucency, and this could also give some kind of a hint on a syndrome or a disease or a condition that is associated with this phenotype. So in the main variant dashboard in the fast track, which is the first tab that we see as default when the analysis is written, the variants are sorted by the relevance of the phenotype in a descending way, which means the first variant was highly relevant or associated with the phenotype in a descending order. So the first variant is a variant in a gene named RERE, R-E-R-E. In this specific location, the reference is A, the alternative is a bit long, and this is, if we go to the effect, this is an in-frame indel. Under the clinical evidence information, we can see that this variant was classified before in ClinVar as conflicting. Oh, sorry. If I just click on the columns, they would be sorted again. Now I want to go to ClinVar information, click on ClinVar, jump to this page just to see what exactly is the conflicting evidences or classifications here. If I scroll down, I would see the different submissions, and I see that one submission by GeneDx is a variant of unknown significance, and there are two additional benign classification by Invita and by CGUT. All three are large and known labs, and we can trust that this is for sure not a likely pathogenic variant. So if we go back to the analysis for sure,
Hi, sorry guys. Yeah, we see that there is an issue. Jade, I think you should unmute yourself. Let's go back a few, like 1 minute back. Jade? Just a second, we'll fix this. Sorry, all Jade, please just unmute yourself and just go 2 minutes back.
I'm so sorry, everybody. See, I was listening to it the whole time by myself, and I didn't think that there was a problem.
Just 2 minutes or 3 minutes back, and we can go again. That's it.
The analysis and other information, including the version set of all the annotation sources, etc. So if I go to the variant dashboard, this is what we will see. Of course, we provided the phenotype in advance, but we can still change that during the analysis. So if you click on the orange bar here and you go to the phenotypes, you can see that we provided the phenotype here is increased nuchal translucency, which is a phenotype in HPO term, which is a well-known in fetuses. We must say that this is highly unspecific. This could be a random increase of the nuchal translucency, and this could also give some kind of a hint on a syndrome or a disease or condition that is associated with this phenotype. So in the main variant dashboard in the fast track, which is the first tab that we see as default when the analysis is open, the variants are sorted by the relevance of the phenotype in a descending way, which means the first variant was highly relevant or associated with the phenotype in a descending order. So the first variant is a variant in a gene named RERE, R-E-R-E, in this specific location. The reference is A, the alternative is a bit long, and this is, if we go to the effect, this is an in-frame indel. Under the clinical evidence information, we can see that this variant was classified before in ClinVar as conflicting. Oh, sorry. If I just click on the columns, it would be sorted again. Now I want to go to ClinVar information, click on ClinVar, jump to this page just to see what exactly is the conflicting evidences or classifications here. Now, if I scroll down, I would see the different submissions, and I see that one submission by GeneDx is a variant of unknown significance, and there are two additional benign classification by Invita and by CGAT. All three are large and known labs, and we can trust that this is for sure not a likely pathogenic variant. So if we go back to the analysis, for sure this variant could be classified as variant of unknown significance and not as a likely pathogenic one, which for me is kind of relaxing for this specific variant. In addition, we can see the NOMAD, or the maximum allele frequency count, and we see that there are 94. If I click on it, you would see exactly the details, but there are 94 in total heterozygous carriers in NOMAD, which for sure remove or excluding the possibility that this is a pathogenic variant in a dominant model. So this was ruled out. The second variant is another variant which received a conflicting evidence by ClinVar of a variant of unknown significance with a likely benign. And this is for sure, or indeed, another VUS variant that is probably not causing disease in an autosomal dominant manner. So another even likely benign, but the specific classification right now is not that important, as we understand that these are not causing variant, disease-causing variants. The third variant is in the gene PTPN11. If I click on that gene, I would see the OMIM inheritance and the exact phenotype that is associated with this gene so far. What we see here is Noonan syndrome. Noonan syndrome is a well-familiar, even common syndrome. I can click to go and see more information, and we will see it shortly in the presentation. What's even more important about this variant is if I go to ClinVar, I would see that this is a pathogenic classified variant, actually with almost dozens submissions, different submissions. It also has a ClinGen variant curation, which is extremely helpful. So in ClinGen, as we described in the previous classes, some of the variants were been through a committee in a thorough classification by the ClinGen team, and this is one of these variants. And I also have the detailed ACMG criteria, which you will learn all about with Dr. Ali Tabish. The pathogenicity of this variant is pathogenic. So for sure, this is a pathogenic variant. ClinGen is a trusty source, and you for sure you can go with its classification. What's interesting about this variant is that if I go to NOMAD, and if I click to see the distribution, I see that there are 3 heterozygous carriers for this specific variant. Now, this kind of gives me a clue about reduced penetrance for this syndrome, for the Noonan syndrome. And also just to remind you that this is a single exome sequencing, but we know that there is positive family history for the older child home that had some kind of a cardiac abnormality. For this specifically provides me a hint that this might be an inherited variant from one of the parents, and that this could repeat in every pregnancy in 50% of the offsprings. So this is a pathogenic variant for sure, as we saw by ClinGen, by ClinVar, and I can see here all these submissions by ClinVar. The phenotype that is relevant is Noonan syndrome. And what's very nice is that there is a very high correlation or association between the phenotype, increased mucosal translucency, and between Noonan syndrome, as this is one of the hallmarks of one of the phenotypes that we well known to see in Noonan syndrome patients during pregnancy. So I can continue and search, but overall we can see that the third variant, as it was sorted by the default filters, the default associations of the phenotype, was the causing variance in this specific for this specific family. Now going back to the presentation, just an overview on Noonan syndrome. So Noonan is a genetic disease with specific facial attributes. We can see here the dysmorphism. There are heart deficiencies, as we saw in the previous child in this family. The affected individuals also have short stature and developmental anomalies. The key features in terms of the dysmorphism would be a wider than standard space between the eyes, low set ears, we can see it here almost, a prominent forehead, and then cardiac features would be kind of diverse, but mainly pulmonary valve stenosis and hypertrophic cardiomyopathy. Both could be hinted by the increase of nuclear translucency. The growth of the patients is they usually have short stature and developmental delays, to a certain degree, could be more, could be less. So this is a variable developmental delays. And sometimes the patients show bleeding issues, which we also saw in the first child in this family. And there are some skeletal anomalies. The inheritance is autosomal dominant inheritance with partial penetrance. I think this is a very important syndrome to know because of the partial penetrance. Because if we did have a trio and we saw the inheritance from one of the parents, at the first second, this could be confusing because you see a healthy parent inheriting a variant in a dominant gene, though, but still the parent is healthy. And this still can be a disease-causing variant. The partial penetrance is very confusing in Noonan syndrome. We see parents that are 100% healthy, and they have affected fetuses, children, and it's a very well-known phenomenon for Noonan syndrome. The genetics of Noonan syndrome, so this is caused by likely pathogenic variants in the RAS-MAPK pathway. Specifically, in this case, we saw PTPN11, but this is not the only gene that is characterized or leading to Noonan syndrome. PTPN11 gene encodes for a protein named SHP2, and this is an essential for RAS-MAPK pathway regulation in cell process, including differentiation, migration, and apoptosis. This is one of the most researched pathway in the human genome or the human cells, with a lot of research around the RAS-MAPK pathway. We know that some of these different proteins in this pathway known to lead to cause Noonan syndrome, when you can see these proteins here in blue, and some of them can cause Leopard syndrome, which is another syndrome that PTPN11 can cause. All these syndromes are one of big family of the RAS-MAPK pathway diseases, and we can see that different proteins in that pathway can lead to the same syndrome. In terms of embryonic development, SHP2, which is a protein coded by PTPN11 is crucial for the heart development, for the blood cells, and for bone development, which makes sense in terms of the well-known phenotype of the syndrome. So pathogenic variants in SHP2 affecting cell function, regulation, and contributing to Noonan syndrome manifestations, as we saw. Specifically for this family, if we go back and think of the genetic counseling, we know that there is, well, we expect that there is an additional recurrence rate of 50% for every offspring. Of course, one of the genetic counseling outputs of the report that was provided for this family was a recommendation to do a segregation analysis to find the inheriting parent, and accordingly, or also to do segregation analysis in the affected sibling here to indeed show that that she is affected with Noonan syndrome as well. That's it. So with that, I want to finish. We can take questions at the end because we don't want to waste another minute before we start with Dr. Ali Tabish lecture. Of course, you can post questions on the chat and we read them live, and we will provide answers at the end of this class. Thank you.
Perfect. I just wanted to say a couple of words here. Do we have a minute, Jane?
Yeah, go ahead.
Okay. I just want to highlight. See, this is a very classical case for, and I'm really happy for this Noonan syndrome, and the patient has knuckle translucency. I just wanted to highlight. See, it is very common syndrome, Noonan syndrome, and it's not easy to pick up clinically. Short stature could be very mild, mitral valve stenosis could be very mild, intellectual disability in the Noonan syndrome could be very mild, and it's really underestimated syndrome. But the point is, see, just one knuckle translucency. Okay, so we clearly have to understand the clinical heterogeneity of the syndrome. The genetic disorders, they present clinically wide spectrum of symptoms, and we have to really understand how those disorders present. And with just like one knuckle translucency, we can identify the relevant variant. This is just, I would like to add this in.
So we'll take more questions at the end. Jade, are you ready with the next lecture? Okay, enjoy.
Go ahead.
Thank you. Thanks, Jade. Thanks for the introduction. Hi, I am Dr. Ali. I am a variant scientist. I will be going through the ACMG guidelines. So this is the first lecture. It's actually the fourth lecture, but it's the first lecture for the ACMG guidelines. I will give you, in the first half of this lecture, I will give you a broader overview, a high-level overview on the ACMG guidelines, and then we will go through ACMG specific rules. So in this first presentation, I will present PVS1, and which is ACMG guidelines, part of ACMG guidelines, and then we will go through the rest of the ACMG guidelines. Okay, as I mentioned, my name is Ali Tabish. I'm senior variant scientist. So in this presentation, I will teach you on ACMG guidelines, ClinGen expert panel guidelines, actually the refinements of the ACMG guidelines, overview of the variant classes, ACMG variant classification, landmark publication, and overview of the PVS1 guideline. So let me start with a typical scenario as a clinician. So usually, or most of the times, the patients we see as clinicians are the referrals. The patients usually coming from the pediatrician, sometimes the internist, or even obstetricians for the antenatal cases. So the patient first presents to the pediatrician, they have a family history, or the patient presents with the developmental delay, which is one of the very common reasons to visit doctors, pediatricians. So they do the first clinical assessment. They will record the patient history, developmental milestones, if there is any speech delays, is there any other relevant features from the patient side. They also sometimes take the family history. Okay, so when a pediatrician taking a family history, then a geneticist, then an internist, there is a slight difference. You will see when a patient comes to the geneticist, the family history is usually more extensive compared to an internist or even pediatrician. And same goes for the dysmorphology, because the pediatricians and internists, they are not well trained for the genetic dysmorphic features. So being a geneticist, we usually have to look for the great details into the dysmorphic feature of the patients. And then there is a lot of different workups already which has been done in the radiology for the lab works, MRI, CT scans, LFTs, RFTs, different lab works has already been done for the patients. And if there's any special investigations from the pediatrician or the internist side, these already have been done. So we compile all these information, create a pedigree of the patient, see if there is any specific inheritance pattern running, either autosomal dominant or recessive or X-linked. We try to understand what is the inheritance of this patient disease. We try to build the differentials, what could be a possible diagnosis of this patient, what kind of genetic test we do need to order. So once we have the complete comprehensive phenotypic assessment, then we decide, depending on the differentials, we decide whether the patient should go for a whole genome sequencing, whole exome sequencing, panels, or other genetic testing. Once this genetic workup has been done, we receive the genetic data as a geneticist. We have the clinical data of the patients, and we implement ACMG guidelines to identify what is the causal variant for this patient phenotype. The reporting process, that could be indication based, like what the patient clinical picture tells us, or the carrier screening for general health assessment, or it could be a secondary finding like late onset cardiomyopathies or the cancers. Okay, ACMG guidelines address mainly Mendelian diseases, the germline inherited variants, and these are single nucleotide variants, SNVs. And there are separate guidelines for large copy number variants, somatic variants, and cancer variants. So ACMG has a separate guidelines for the SNVs and separate guidelines for CNVs. So this is a landmark publication by Richard in 2015, which deals with ACMG variant classification. There has been previous publications by ACMG, but this is the one which is a landmark, which we use almost every day. And all the different revisions and refinements are based on these publications. And I highly recommend everyone to go and try to read and understand these ACMG guidelines from this article. Okay, next, sorry. So after this 2015 ACMG guidelines by Richard, there has been updates and revision. So this is what the one on the top is 2021, the technical standard guidelines, molecular genetic testing for ultra rare disorders. So this is one major revision. And then there are several specific revisions. For example, there's the ACMG vector resource genetic evaluation for short stature. So patient presenting with a short stature, there's a revisions for short stature patients. There's a revision for CFTR causing cystic fibrosis. And also there is a revisions for X-linked disorders. And I highly recommend everyone to consult these revisions as well. And there are several others which I haven't put into this slide. There are several other revisions by the ACMG committee. Okay, so why do we need ACMG guidelines? As I said, the variant interpretation and classification, it's been going on since the NGS, but not in a standardized way. So ACMG guidelines gives us a standardized process. Here I give you an example. One variant. This variant is in the HFE gene, which causes hereditary hemochromatosis type 1. And if you type this variant into ClinVar, you see conflicting classification of pathogenicity. Some labs have classified as a pathogenic, some they have classified as pathogenic with a low penetrance, as a variant of uncertain significance, and even benign. So you see, if this variant is pathogenic, it cannot be benign. If it is benign, it cannot be pathogenic. So there is clear need to have a standardized way of classification, and that's been addressed by ACMG. Okay, and then after ACMG, what is ClinGen? Here is a link in the bottom of this page. If you go and search ClinGen into the Google, you will get straight to the ClinGen website. You can also copy paste and then try to go to the ClinGen website. So ClinGen gives us a refinement on ACMG guidelines, the 2015 original publications by Richard. So this ClinGen gives a lot of different refinements on how these rules have evolved over the period of time since 2015. So if you go into this, there are several tools and resources. There is a criteria specification registry, so if you click on that, you will see good details on different criterias. There is a variant pathogenicity tools. There is refinement for PBS1, for example, this highlighted one. There is a refinement for benign standalones. We will go through our later in this presentation PBS1 and different other criteria. But just to give you an idea, the ClinGen gives a refinement of the original Richard ACMG guidelines. Again, there's a refinement on reputable source criteria, PS3, BS3 refinement, PP3, and segregation PP1. You can also find detailed documentation for these refinement, as I highlight one here, and there are several others. You can go, there's a PDF available, you can download, you can read, and also there are refinements on indications. For example, here I highlighted like ClinGen cerebral carnitine deficiency guidelines. Also, there is ClinGen refinements for the cystic fibrosis and other disorders like seriopathies and cardiomyopathies and others. So we highly recommend you guys should go and not only the Richard original publications, but also the ClinGen expert panels, try to understand, try to learn their guidelines, their refinements on the Richard guidelines. Okay, so according to ACMG variant classification, variant classes, there are 5 different classes proposed. Actually, this is the 5th one. This is likely benign, and then the 5th one is a benign. So the first one is a pathogenic, then the second is a likely pathogenic, a variant of uncertain significance, likely benign, and the 5th is benign. So there's a typo error. So these are the 5 different classes of the variants proposed in the Richard 2015 paper. So the pathogenic variants are the ones where we have a 99% certainty, this statistical evidence that this variant leads to this disorder. Okay, then there is a likely pathogenic where we have a more than 90% certainty, the more than 90% likelihood that this variant will lead to the disease. On the other end of the spectrum, we have a benign variants where we are sure and we have a high certainty, a likelihood that this variant does not cause the disease. And these are the benign variant. And then there is a likely benign variant. In between these two extremes, there is a gray area, there is a gray zone where we don't have enough statistical evidence, or we don't have enough clinical or functional evidence to classify those variants either into pathogenic or benign class. And these are the variant of uncertain significance. These are uncertain because currently there is not enough publications and not enough clinical or functional data to classify into either extremes, either pathogenic or likely pathogenic. So here's a list, complete list of the Richard ACMG guidelines. There is a 16 pathogenic criteria, the one on the red and different shades of the red. And then there is 12 benign criteria. So the first one is a PVS1, which deals with the loss of function. And it's a PVS, it's very strong criteria. P stands for pathogenic, very strong. 1. This number doesn't give much information, but these numbers were used into the ACMG Richard paper. So the PVS1 is a very strong criteria, and it deals with the loss of function variants. Then there is a strong criteria, PS1, PS2, PS3, and PS4. So the P stands for, again, pathogenic, S for strong, and 1, 2, 3, 4 different numbers given. So PS1 deals with the amino acid changes, the missense, not only the missense, all the same amino acid change as the pathogenic variant, the missense pathogenic variants. Then PS2, de novo. PS3 deals with the functional studies. PS4, the number of counts for the affected individuals. Then there is a moderate criteria, M stands for the moderate, 2, 3, and so on till 6. And finally, in the pathogenic side, there's a sporting, P for the sporting pathogenic criteria, and that deals with the co-segregation in silico and so on, the phenotypic specificity and reputable source. And on the other hand, there is a benign criteria, the benign stand alone. If pathogenic, if a variant has a more than 5% frequency in the healthy populations, that we apply BA1, the benign standalone. And then there's a benign criteria, BS1, where there's a higher minor allele frequency than the disease prevalence. Then BS2 deals with the penetrance and observed in the healthy individuals. BS3, benign criteria extracted from the functional studies, and BS4, the lack of segregation for the benign criteria. And then there is a sporting evidence for the benign criteria. There's a from P1 to P7 sporting criteria for the benign. Like in silico evidence doesn't support the pathogenicity, then we apply BP4, for example. Same goes BP7, the synonymous variants, in case they don't have any splicing effect, we apply BP7. And then there is a guidelines how we can combine different rules. So we have several different pathogenic rules, then we have a different benign rules, and then how we can combine both pathogenic and benign rules to classify a variant. For example, PVS1, very strong. We need at least one sporting criteria to classify a variant with PVS1 to classify them as likely pathogenic. For example, PVS1 with plus PM2. That's a moderate sporting criteria for the pathogenicity. We can apply both to classify a variant as likely pathogenic. And also, if we don't have a PVS1 strong, we have only the strong criteria like PS1, PS2, PS3, and PS4. We can consider at least 2 strong criteria to classify the variant as pathogenic. Or if we have only one strong criteria, not two, but only one strong criteria, then we need additionally moderate criteria, 3 moderate, 2 moderate plus 2 sporting, and so on. So this is from the same publication, and you are highly encouraged to go into this article and try to read how we can combine different rules, the pathogenic and benign rules, wherever they are applicable, and classify these variants. And then likely pathogenic, as I explained just now, that for likely pathogenic PVS1 with just a moderate criteria, one moderate, we can apply, gets to a likely pathogenic classification. And if we have a strong criteria, then we need 1, 2 moderate criterias, or 1 strong and 2 supporting criterias, and so on. We can combine different rules to classify to a likely pathogenic. And then comes to the benign, like standalone. If the variant have a more than 5% frequency in the healthy population, we can apply BA1. And then strong criterias in the benign side, we can apply BS1 or BS4. If there are 2 strong criterias, we can apply like BS1 and then BS2. It will leads to a benign classification. And then similarly, we can combine different rules to get a likely benign classification. So it seems like there's a lot of rules, a lot of different ways we can combine them, and it can cause us kind of complexity. There are several different publicly available pathogenicity calculators. These are not fully automated. They are semi-automated calculators. So these calculators, they can apply some of the rules, like if a variant is loss of function, they can apply, for example, PVS1, or if there is a missense variant with the high in silico prediction score, they can apply PP3. So these are the criteria. Some of these criteria, they can be auto-populated, means the software can apply itself. And there are some evidence where curator has to intervene. For example, if the case is like having a very specific phenotype, we can apply PP4. And we can also search for the literature. If this variant has been published in different functional studies, what are different available evidence for the pathogenicity of this variant from the functional data? Is there any similar clinical data available, like PP1 disaggregation data? So we can also intervene and try to find additional evidence in the process of variant classification. So there is one calculator from the ClinGen. You can go into this website and try to curate the variants and try to learn how it works. And there is also another one from the University of Maryland. You can use that. These are freely available. This is the one from the Genix. So different rules are applied, auto-populated, for example, PM2, depending on if the variant is rare. And this is a loss of function variant, so the Genix calculator software applied PVS1. So it comes to a final classification, likely pathogenic, PVS1 plus PM2. And these are the ones which are crossed. These are not met. So, but we can also search additional evidence and try to implement, for example, PS3. And right now, there is no mark on PS3. We can go into the publication and try to see if there is evidence available, functional evidence, and then we can apply PS3. And it will change the classification to pathogenic, for example. Okay, so this was a little background on what is ACMG, why we need ACMG, and the refinement by ClinGen. So I really recommend all of you to go and this original article from the Richard and read this, try to understand different criterias, and then go into the ClinGen and try to see what is the difference from the original publication and from the ClinGen and how they correlate together. Okay, so next half an hour, we will go through PVS1. So there was original publication from the Richard 2015 on PVS1, which deals with the loss of function variants. And then there is a revision on PVS1, the recommendation for interpreting loss of function PVS1, ACMG AMP variant criteria in 2018. Again, I will recommend to go through this publication and try to understand the PVS1. As I said, this is the pathogenic and benign criteria from the Richard guidelines. As I said, PVS1 is a very strong pathogenic criteria, and we have to deal it very carefully because it will change the diagnosis of the patient. If we apply PVS1, we need at least one moderate supporting criteria to move the classification of this variant from variant of uncertain significance to higher, to likely pathogenic and higher ones. So we really have to be very careful applying PVS1, and that's one of the reasons there is a huge debate and literature available on PVS1, how to apply, where to apply, which genes to apply, which variants to apply. So I really highly recommend to read more about PVS1, loss of function, and other associated material with the PVS1. So the PVS1, the variants with predictable high impact on a gene product functions. So these are the nonsense, frame shift, canonical like +1, +2, splice sites, and the initiation codon variants in a gene where loss of function is an established disease mechanism. So that is highlighted. These variants, these nonsense, frame shift, canonical, splice site variants in a gene where loss of function is established disease mechanism. Okay, so as I said, PVS1 with any other criteria like a moderate PM2, it will go to likely pathogenic variant classification. PVS1 with PP3, PP2, it will go to pathogenic. So just to highlight, this is very strong criteria. Any combination with any other pathogenic criteria that will change the classification from variant of uncertain significance to likely pathogenic or higher. So this has to be very carefully met. Not all the variants resulting in loss of function have equal strength, and that is discussed in new revision. That if a frame shift variance is in the start of the gene, middle of the gene, or end of the gene, or also to look for the gene causality, how well established the gene for that phenotype. Considering all these parameters, it's not that all the loss of function variants will have the same strength. So as I said, the variant types in the loss of function could be nonsense, frame shift, canonical splice site, initiation codon, deletions. We have to take that into consideration, and then variant effect like location, how this variant affecting these exons, is it included or excluded, or also the gene structure and regulation, the alternative transcripts, and the biological relevance of those alternative transcripts, and how this help in the regulation by nonsense mediated decay and the mechanism of pathogenicity. We have to take that mechanisms into the consideration, and then finally decide if we can apply PVS1 like a very strong or at a strong level, or PVS1 as a moderate, or even PVS1 as a supportive level. So it's not like if there's a loss of function variant, we apply PVS1. No, we have to really understand the variant type and the effect at the variant features, and take all these evidence into the consideration, and then apply PVS1 depending on either it is very strong or different level of pathogenicity. Okay, so the next is the decision-making pathways, stop gain, and frame shift. So here I would like to emphasize loss of function, like PVS1 deals with the loss of function variants. And loss of function disease mechanism implies either the absence of a gene product or the synthesis of a non-functional gene product, or gene product with reduced functionality. So what it's essentially saying, loss of function is a disease mechanism. It's not a variant type. So variant type could be frame shift or splice site or any other like loss of function. But this is a variant type. It's not a loss of function. Loss of function is a disease mechanism. So if you have these variants like nonsense, frame shift, or canonical splice site, which presumably will be applied for the PVS1 strong criteria. Now you can apply these either into the homozygous, like when both alleles are disturbed, like if this variant is in both alleles, from here it shows as a P1 and P2, or it is in single allele as a heterozygous. And depending on how this zygosity is, it can yield into a null variants or a haploinsufficiency, or like hypomorphic variants or hypomorphic alleles. Apart from these variants, this nonsense and frameshift variants, there are other types of variants which can also lead to loss of function, like missense variants in frames, synonymous, and both in heterozygous or homozygous fashion. But loss of function is reserved with PVS1. You can apply for the missense if there is a functional studies or additional evidence available for those in-frame deletions or missense affecting a splice site. If there is a functional studies available, we can apply PS3, which deals with a functional study, which we will talk about in the next lecture, PS3, the functional studies. But the PVS1 is only for the loss of function, nonsense, frameshift, chronicle, these variants, not for the missense variants. And as I said, we can apply this to autosomal recessive disorders, autosomal dominant disorders, which leads to loss of function or haploinsufficiency, and also the X-linked disorders. Here in the X-linked, due to X chromosome inactivation, the phenotype can have a spectrum. There's an X chromosome inactivation, and the phenotype, they really could be very severe or it could be very mild. So here is from this article, there's a flow chart for nonsense or frameshift variants, loss of function variants. I will start with this nonsense and frameshift. So we have to look whether if this variant is predicted to undergo NMD. NMD stands for nonsense mediated decay. If we say, yes, this will lead to nonsense mediated decay, then we have to see this exon is present in biologically relevant transcript. If yes, okay, we can apply PVS1. And if we see this exon is absent from biologically relevant transcripts, then we cannot apply PVS1, even though if it is a frameshift variant, but it's not present in biologically relevant transcript, so we will not apply PVS1. And again, next, this nonsense or frameshift variants not predicted to undergo nonsense mediated decay. Then we have to check this truncated region is critical to the protein function. Okay, I will go through where we can see if this information, like if this region of the protein is critical to the protein function. If we decide, yes, this variant affecting, this loss of function variant is affecting a critical part of the protein, then we will apply PVS1 strong, even though it's not undergoing NMD. On the other hand, if the role of the region, the protein region, is not clear or is unknown, then we see loss of function variant in this exon are frequent in general population, and the exon is absent from biologically relevant transcript. So if there is a loss of function variance, it's frequent in general population. So this gives us a hint, maybe this is not a disease causing, or it's not present in biologically relevant transcript. Again, it hints us it's not a disease causing. So we will not apply PVS1. But if the loss of function variant in these exons are not frequent in general population, and the exons are present in the biologically relevant transcript, then we again see, we are talking in the context that it doesn't lead to NMD. Then we see the variant removes more than 10% of the proteins, then we will apply PVS1 strong. And if variant does not lead to more than 10% of the protein defect, we will apply PVS1 strong. So I will highly recommend just go in this article and try to understand how we can apply PVS1, because it's a strong criteria. As soon as we invoke PVS1 with any other criteria, it will change the variant classification. So here we know there's how this cells make a mRNA from transcriptions. There's a ATG start codon, there's exon, there's intron and stop codon, and those are linked together in the process of transcription, and then these are translated into a protein. So if there is a PTC, premature termination codon, like a loss of function variant, it will really disturb this process, and it will activate NMD, nonsense mediated decay, that will leads to decay of this product from this gene. And a transcript is predicted to escape from NMD. So NMD will not happen if the premature stop codon is localized into the last exon. If this loss of function is in the last exon of the gene, NMD will not activate. There will not be a NMD process activation in the cells if it's the last, or it's a 50 to 55 base pair of the penultimate exons, so 1 exons before the last exon. So if it is in that range, 50 to 50 base pair from the penultimate exons, again, the NMD will not happen. Or this gene is a single exon. There is no, sorry, I have to. If this is a single exon coding gene, or the premature stop codon is localized within the first 100 base pair of coding sequence, like start proximal NMD insensitivity. So if these conditions are met, then there will be no nonsense mediated decay. And next, how we can evaluate all these, like I mentioned, biologically relevant transcript, biologically relevant protein region, how we evaluate this information. So there is a direct evidence or indirect evidence. Direct evidence, very simple, it has to be published. Functional evidence represents the main criteria to establish the relevance of an exon or a transcript, like, for example, DMD. The transcript, the most relevant transcript, the biologically relevant is this one, the one mentioned here. And then comes to the indirect evidence. So indirect evidence, we take it from the different databases, for example, localization of a pathogenic variant within the exons. So we can get this information from HGMD, or we can go into the ClinVar database and see localization of other pathogenic variants in these transcripts, in these exons from the clinical cases, and the frequency of the variant resulting in loss of function in exon in general population, we can get it from GenomeMed, sorry. So we can go into NOMAD and see what is the frequency of loss of function variants in this gene in healthy population. Or we can also get information, alternative transcription of the exon. Also, we can get the GTEx database. We can get some information from GTEx database of if this exon is a part of this transcript or not. And finally, there are different gene-specific databases available. For example, TTN database. So if there's a loss of function variant in a specific part of the TTN gene, does it leads to nonsense mediated decay or not? We can decide, go into this website, CardioDP, and see if this variant affecting which transcript, which part of the Titin gene. Either you can go to the A band or I band or different other bands, and then apply PBS1 accordingly. So here is NOMAD database, which collects information from different other databases like ClinVar, GTEx, Ensemble, Genome Browser. It can be used as a main source of the indirect evidence to evaluate the biological relevance of the transcript. So if you click on the hide transcript, if you click there or show transcript in default, you will see what are other transcripts available for this gene. And you can decide, okay, if this variant is in biologically relevant transcript or not, or biologically relevant exons or not. And then you can also get some more information from GTEC expression data sets. See, in this exon is not part of, are very little expressed in this transcript. So if a loss of function variant in this exon, but this exon is not being incorporated into this transcript already, so the loss of function variant will not have that pronounced, will not have that effect if it is in this variant which is already being excluded from this transcript. And then you can get more pathogenic or likely pathogenic variant information from ClinVar. If you click on this here in ClinVar PLP section, you can get what is the frequency of likely pathogenic variants in ClinVar for this gene, and you can decide based on this one, the biological relevance of the transcript. And also in the healthy population, what is the, if you click on the PLOF only, you can also get a list of the variants in the healthy population from NOMAD. Also, there are other parameters like PLI, probability of loss of function constraints. They give a lot of information about if the gene is loss of function tolerant or intolerant. You can go to the ClinGen and try to identify what is the haploinsufficiency score for this gene. So these are different resources where you can get more information about loss of function, and if these criteria are met, we can apply PVS1. And how we can evaluate the functions of a truncated region. So for this, we can go to a UniProt database and try to identify the variant location into the which domain of this protein. And according to that, we can decide if this variant will have a significant effect, on the protein domain. And this UniProt dataset will give us more information on a functional role of the different protein domains. And also we can get localization of non-truncating pathogenic variants within the exon or downstream of the localization of the variant. And this information we can go from HGMD or ClinVar database. For example, if you just go and click on the BRCA1, and you will see a list of all the non-truncating variants in the BRCA1 gene. And this is guidelines on how we can apply or navigate through different strength criteria of PVS1. The final decision regarding to the PVS1 strength level should take in consideration the strength of evidence supporting the loss of function disease mechanism for a relevant gene disease association. So it's not just like the variant is loss of function. We have to see if the gene has a loss of function mechanism. So it goes like a flow PVS1 flowchart. If this we follow the PVS1 flowchart. Clinically validity classification of a gene is strong or deaf. That means the gene is well established to be loss of function. And the gene disease association is well established. Sorry, I'm mixing up two different things. So the gene disease association is well established, and then there are 3 or more loss of function variants as pathogenic without PVS1, and more than 10% of the variants associated with a phenotype are loss of function across more than one exon. So if these criteria are met, we can have a considerable statistical evidence confidence that we can apply PVS1. But we should decrease the final strength by 1 level, from, for example, from very strong to strong, if clinical validity classification of a gene is at least moderate, and 2 or more loss of function variants have previously been associated with the phenotype across 1 different exons, and there is a animal model which recapitulates the disease phenotype. So there's like 2 or more than 2 loss of function variants and the disease model. And we have to decrease the final strength even 1 more, like 2 levels, from very strong to moderate, if the clinical validity classification is at least moderate, and either there are 2 or more loss of function variants, or there is an animal model recapitulating the disease phenotype. But if these criteria are not met, if there is no evidence that loss of function variants cause the disease for this gene, we shouldn't apply PBS1. And we can, as I said, we can get this information, some of these haploinsufficiency we can get from ClinGen, from Decipher. We can get a PLI, the constraints, the loss of function constraint information for every gene from the NOMAD. So this is just one, I think this is the last slide, I presume, yes, last slide. So this is example of loss of function variants in BRCA1. So if you go into the ClinGen, you just type like BRCA1 and then select short variants like SNVs, less than 50 base pairs, and then select frame shift variants, nonsense, and splice site, and then classification likely pathogenic and pathogenic. You will get a list of BRCA1 variants which are loss of function, likely pathogenic, and pathogenic. And this is just an overview how these variants are scattered all over the genes on the BRCA1. And this is a list of these variants. I think you already have a good overview. You already have learned a lot of information about ClinVar and how to use a ClinVar. So once you go through the ClinVar for the BRCA1, you will see the list of the loss of function variants where we can apply PBS1. And I think this is the last slide. Yes, and thank you for your participation, and we will answer your questions. Thanks a lot.
Thank you, Ali. That was very informative. We would like to go on to the question and answer part of the lecture. And we look forward to hearing what Hagar and Ali have got to say.
Hi, I have a list of questions. So, sorry. Someone asked about ACMG secondary findings. So there is a recent publication, 2023, where they have 2.3 version of ACMG secondary findings. And I believe there is more than 80 genes. And you always consult latest guidelines for the secondary findings. But one important thing, we always check before we write any patient report, we always check if they ask for the secondary findings, if there is a consent from the patient to include the secondary finding or not. If there is, we consult the recent guidelines, which is published by ACMG in 2023, and then we report accordingly. There was a question about the mission of ACMG. So the mission of ACMG is very clear: to standardize clinical genetics reporting. Okay, as I mentioned in the presentation, and even if you go into the ClinGen and type BRCA1, and you navigate through different variants in the BRCA1, you will see a variant is classified likely pathogenic by some labs and different classification by some other labs. So there is no coherent classification in ClinGen, right? So ACMG is trying to standardize this reporting of the genetic variants and developing different rules and guidelines to classify genetic variants. And of course, there is a different groups in ClinGen which are refining these guidelines. That's the message, the functions of the ACMG and ClinGen groups. There was a question about PS1 and PM5. We use ClinVar. So for PS1, we need to get clear evidence. Okay, it has to be very clearly known for PS1. PM5, yes, we can apply PM5 based on the ClinVar evidence, but it's really not encouraged. So I recommend every time go into the publication, try to find independent source, a verifiable source. Okay, different labs they say, okay, this variant is de novo, but there is no way you can verify. So I always recommend go into the publication, try to find what additional evidence you have for PM5. If you get evidence in publication, yes, you can apply PM5. And there is a question about gain of function. Can we apply ACMG guidelines for gain of function variants? Yes, we can apply loss of function, gain of function, dominant negative, all these mechanisms. We can apply ACMG guidelines. And there are a couple of more questions. Can you suggest an efficient process to guide our search? I presume you mean guide our search. You mean how we find publications or variant of interest? I think there is a very good tool Dr. Hagar mentioned in one of her previous lecture called Litvar. So this is excellent tool. Always go to the Litvar, and that will really help you find the variant of interest. Other ways, going to the PubMed, doing a manual search, or even Google Scholar can help. But that takes a time, and Litvar really helps a lot. Every day, it helps us in finding the variant of interest. And there is another question, again, the same. Should we use ClinVar or not? Okay, ClinVar was recommended in the original ACMG guidelines as a reputable source. So we use PP5. If a variant is classified as, for example, a CFTR variant is classified as pathogenic in ClinVar, in the original guidelines, yes, we will use it as evidence for PP5. PP5 is a pathogenic, sporting pathogenic criteria. We will apply PP5, but with the recent guidelines and refinement from ClinGen, use of PP5 is excluded. We don't use PP5 anymore. And the reason is we are overcounting the pathogenic strength. We are using the same evidence and overcalculating the strength of the evidence. And also, ClinVar is not really, anyone can make an account and they can submit the variants without any stringent quality control criteria. So these are some of the reasons the recent guidelines, they recommend do not use PP5 as a pathogenic criteria. And there is a last question: how to take decision on biologically relevant transcript? So as I mentioned, as I replied in the message, NOMAD is the main source. So if a loss of function variant is in an exon, which is not a part of the transcript, we will not apply PBS1. And we can get this information from GTEx data, expression data sets, if this exon is a part of the main transcript. And also, you can go into the NOMAD and try to see likely pathogenic variant in that transcript. If there is a tons of likely pathogenic or pathogenic variant, loss of function variant in that transcript, in that specific exons, then you might get an understanding, okay, this might not be a relevant transcript. There is a healthy people having a loss of function variants in this in these exons. Maybe this exon is not incorporated into the main transcript. This exon. Also, you can get you can get information from Decipher, this information, and you can also check more information from the ClinVar as well. I said ClinVar is, use it with caution. If you try to find there is a missense pathogenic variant in an exon of your interest, you really have to see if that missense variant is, you have to classify that missense variant first. If it is really pathogenic, you can take that evidence from ClinVar. Okay, and then you can decide, okay, if this exon is deleted in this gene, then you can, this is also another evidence you can apply PVS1. So you have to get information from all these different sources, because PVS1 is a strong, very strong pathogenic criteria. If you apply a PVS1 to any variant like frameshift or loss of function or splicing variants, it will change the classification of the variants. So I recommend take information from all these sources, all these databases, and then aggregate all the evidence and then take the decision.
Thank you, Ali. I want to jump in just for homework information, because there are a lot of questions regarding the homework. So we just posted a file with all the results, with all the variants, the pathogenic or disease-causing variants in these cases. It's in the same OneDrive that was shared with you. You do not need to submit the homework. This is for you to test. So this is a self-test analysis. We hope that this was fruitful, and we understand that some of the individuals or the participants could not access the link. This is most probably due to regulations, security regulations, and access to Microsoft. We have another source for the data. So if you still have any issue with the homework, or you cannot upload a case or anything, just please write to support. We will try to help you. That's it. You're welcome to go online and check if you got the cases solved correctly.
Congrats, everyone. I see a lot of emails, a lot of people asking various questions, trying to get the work correct. And we've all learned a lot from Hagar and Ali. And thanks for this lecture. And we look forward to next week.